大家好，欢迎大家来到 CGM 第86期学术报告，我是今天的主持狄晨露，很高兴邀请到 James 邹老师来做报告。今天的报告就像 James 刚刚说的，以英文为主，然后回答时间可以用中文。下面我简单介绍一下 James. James is an associate professor of biomedical data science and faculty of computer science and electrical engineering at Stanford University. James received his PhD from Harvard in 2014 and was a member of Microsoft Research. Before this, he completed Math Part 3 at University of Cambridge and was a Simmons Fellow at UC Berkeley. He joined Stanford in fall 2016, and he is a faculty director of the new university-wide AI for Health program. He works on a wide range of problems in machine learning, from providing mathematical properties to building large-scale algorithms, and he's especially interested in applications in genomics and computational health. He's going to talk about his recent work. Let's welcome James.
Okay, thank you very much, and thank you for the invitation, for organizing this this presentation. And I hope that everyone's staying safe and healthy during the time of the virus. Yeah, so I will tell you about some of the projects we've been doing the last year and 2 years on applying machine learning algorithms for improving the safety of genome editing. And I'll also tell you, so that'll be the first part. I'll also tell you about some techniques that we've developed that I think will make it easier to understand what these deep learning algorithms are doing, that I hope to share with you some of these techniques. Okay, and this is my students, my postdocs, my group here at Stanford. So just tell you a little bit about my group and also our interests. So we are quite excited about developing new machine learning algorithms for a diverse set of medical applications, especially applications that relates to human disease and human health, right? And the kinds of applications we look at tries to look at human disease, human health from quite different perspectives, right? So we look at it from the perspective looking at molecular data, also from looking at cellular data, which also could include imagings, as well as looking at it from like medical records, from devices and cell phones. Right? So today I'll tell you actually a bit about some of our work on the molecular side of genome editing. So many of you probably have been quite familiar with genome editing, right? And you probably have also used genome editing CRISPR technologies in some of your research. So the community is really excited about the potential of these editing tools, right? And the ability to make precise edits, especially in genomes of humans, and can lead to new diagnosis, new treatments, new drugs, right? So that's actually really exciting. However, even though there's a lot of excitement, right? So one of the big challenges in the field of genome editing is that we have made a lot of progress on the experimental side, but a lot of these tools are really new, right? And we don't really understand, and we don't understand them very well of how exactly these tools act on different cells and on different genomes, right? And that becomes especially challenging when these tools for editing are actually deployed for therapeutics, for diagnosis in humans. Right? So a big part of our research in my group is to develop and use machine learning to really improve our understanding of genome editing, and also to improve our ability to control and to optimize these editing tools. Right, so if you have any questions about this, feel free to interrupt me now or ask me after the presentation. So with that, I thought what I'll do is just to bring everyone up to speed, right, by giving you like a very quick primer of how genome editing works, and then dive into more details about what are the machine learning challenges. Right, so in a nutshell, this is basically, when we talk about genome editing, we're talking about taking these little molecular scissors, which are this little, this blue object here, right? So this is the CRISPR-Cas9 system, and we can target it to a particular location in the genome, and it goes to that location, right? It latches onto the genome, sort of like zippers, and it actually breaks the genome at these two places, like shown here, right? And it creates a double-strand break. And the important thing is that after the genome is broken, right, after this break, it's sort of, the genome sort of tries to heal itself, right? It wants to stitch itself back together, and does that by essentially doing sort of like a random walk, right? It's trying to find some combination of nucleotides, ACTGs, that are floating around so that it finds the right combination to put the sort of stitch itself back together. And the important thing to note is that this process, which is called DNA repair, right, where the genome tries to stitch itself back together, is very much a random process, right? So that means that if I actually do this in 2 different cells, right, I could actually get 2 different outcomes because of the randomness involved, right? So sometimes I'll get an A inserted, other times I'll get a T inserted, other times I might even get deletions. So this actually is one of the key computational challenges, right? Because this random process could introduce insertions, deletions, mutations that are unexpected. And that could be problematic, especially if we're trying to really control the precision of the genome editing, right? So for example, if you're trying to apply this actually for therapeutic purposes in the human cells, then you want to make sure that you don't introduce unexpected mutations, right? You want this process to be as reliable, as robust, as precise as possible. And this is where we think that machine learning can become really helpful. Right? So I'll tell you about this recent paper that we published last year, where we try to develop machine learning algorithms to really try to understand and to model and to predict this DNA repair process that's introduced to genome editing. Right? So more precisely, the machine learning problem is the follows, right? So, the engineer or the biologist says, okay, so here's a particular location in genome, right, that we want to make the edits. And we would like to predict, using some features of the DNA near that cut site, what are the likely outcomes? What are the likely insertions, deletions, and mutations that are likely to happen if we make this edit at this location? Okay. So the ML problem is then this prediction problem, where we take input the DNA sequence features and try to predict the outcome. And just to place this into a context of how does this relate to broader questions related to CRISPR? Right? So in my view, there are several, I made a list of several what I think are the key types of questions that relates to computation in CRISPR and in genome editing. Right? So the first type of question is looking at these on-target effects. Right? So what this means is that at the location where we're making this edit, right, when we make this cut, what are the likely mutations, insertions, deletions when I make the cut at this location? Right, so these are on target because we're looking at the effects at the place where we're targeting. This is, I think, probably the most important type of problems when we talk about CRISPR, and this is sort of the problem that we're trying to tackle here with our machine learning model. In addition to on-target effects, I think it's also quite interesting to look at what are the off-target effects. Right? So that means is that instead of looking at the part of the genome you're targeting, maybe you're targeting actually remaining parts of the genome. And then we want to know, okay, so what are the accidental mutations at the rest of the genome? Right? So these are off-target effects, which is also very important. And we've been doing some works there, but I'll focus more on the on-target effects. There are also really interesting questions around analyzing the CRISPR screens, pull screens. Right? So we have some papers for analyzing CRISPR screens. I'm happy to discuss that more at the end if there's interest. Okay, so how do we actually go about trying to solve this problem? So in order to really be able to have a model that predicts on-target CRISPR outcomes, right, so we first need to generate, I think, a nice large data set. Right. So we actually worked very closely with our collaborators at the Shenzhen BioHub, where I am also an investigator, to generate, I think, a really nice data set that we've also made publicly available. So the data set comes from basically doing a lot of the types of following experiments. Right. So for every experiment, right, so we have a particular target, and in that target, right, so we'll have a CRISPR that goes there to make that cut, like we saw in the video. And after you make that cut, we'll actually do very deep sequencing at that location. Right? So we'll generate around like 60,000 reads on average at this location So you can view each of the reads as basically sort of like a IAD sample from this population of cells to give you a sense of what outcomes, what insertions and deletions are likely at this location. So in this table I've shown here in this figure, it just gives you some of the example outcomes at this particular location, right, at this particular target site. So for example, you see, so each row here corresponds to one type of outcome. Here I'm just showing you the most common rows for the most common outcomes at this location. So the first row actually corresponds to the events where there's actually no edit, right? So that means that in those cells, the CRISPR goes in and for various reasons, either didn't cut or it cut, but actually went back to the original sequence, so there's no edit. And then you also see here in these two columns, right, the number of reads, which corresponds to roughly the number of cells where that event happened. Then, in addition to these no edit, no variant events, right, so now you actually also see additional events, outcomes, where you do have insertions and deletions. And so for example, the fourth row here, that corresponds to these 3 nucleotides in the middle that are actually deleted, right? So they're missing, and that's actually quite interesting though. And some of the later rows corresponds to when larger chunks of sequences are being deleted. Great. Is the data clear? Any questions so far? Okay, and is it also clear what's the problem that we're trying to solve? Right, so the problem we're trying to solve is basically I want to take some features about this target sequence, and we want to basically able to try to predict information about all of these different outcomes. Okay. So to generate the training data, right, so we do this for every location where we make the cut, we do this deep sequencing. And then we do this deep sequencing, not at just one location, but across many locations across the genome, right? So we did this in over 1,500 locations in T cells, and we use T cells because those are the cells that are therapeutically oftentimes the most important and most relevant. And then we also actually repeated this across different individuals. Right, so that's actually quite important to get a sense of how much variation is there actually across different individuals because they have different genetic backgrounds. And then I'll also tell you later on how we actually validate this in several additional cell types. Right, so we perform similar kinds of experiments in other cell types beyond T cells as additional validation. So there's actually quite a large set of data that's being generated. Okay. So just to explain a little bit more about what the algorithm is trying to do, right? So it takes this input, different features, right, about the DNA sequence, right? So this could be features like the ACTG content itself. It could incorporate features about, for example, the thermodynamics, the energy, or the chromatin status, right, of that DNA sequence. So those are the input features. So the output of the model, what the model is trying to predict, are the following quantities, right? So we're trying to predict if I make a cut at this location, what fraction of the outcomes, right, do I see insertions versus deletions, right? So what that means is that if I do this, let's say in 100 cells, right, how many cells will actually have insertions and how many cells will have deletions? If it has insertions, right, what is the average typical length of these insertions? Or what's the length of the deletions? And which nucleotides actually gets inserted, right? So these are all quantities that we want to predict, and that would actually enable us to think about designing better CRISPR and safer CRISPRs. Okay, so there's a variety of ways that we can develop algorithms for doing this, right? So we have explored algorithms both based on neural networks, also based on random forests and decision trees, right? So I'll come back to the details of the algorithms, if you are interested in the end. But I think we tried a lot of different models to try to make these predictions. But I think it's actually maybe more interesting to think about how one might actually validate this kind of algorithm and how one might use it in practice. Okay, so I think when we think about applying machine learning in genomics, especially in genome editing, it's extremely important to think about how one actually, what's the right way to validate and to test these models. Okay, so the first way that we went to test these models is by doing something that people typically do in machine learning, which is to have a held out data set, right, from these T cells, and then just to have the model trained on the remaining data and then just test it on the held out data. Right. And here I'm showing you the results from these 3 different tasks for predicting precision, frame shift, right? So how often are we likely to have a frame shift due to edit at a particular location? Also, how likely are we to have insertions versus deletions, right? So the model that we developed, which is called Sprout, right? So it actually achieves reasonably good accuracy for each of these 3 tasks, right? And it's certainly substantially more accurate compared to a couple of other methods that people recently developed. Okay, so that's the baseline for validation, but I think that's actually not enough, right? To really ensure the quality of these predictive models, what we did next is to actually go out in a separate experiment that's performed by different people later on, right? So we actually collected new T cells with new locations, right? Locations that have not been seen before, right, in the genome by the algorithm. And then we tested the performance of the algorithm without any modification on these new T cells and on these new locations, right? And it's actually very promising that they give essentially very consistent performance for Sprout. Okay, and then even that, I think it's a good step, but it's not quite enough, right? Because we want to be able to show that the algorithm works really well beyond just one cell type, beyond T cells. Right, so what we did was actually to look at other cell types, including iPSCs, which are sort of induced pluripotent stem cells, which is also really important for therapeutic applications. And notice that the algorithm was only trained on T cell data, so it had actually never encountered iPSCs before. Right? And then again, when we applied the algorithm without any tuning to iPSCs, it also actually give quite reliable and quite strong performance. Right? So there's some decrease in performance, decrease in accuracy, because now it's looking at a different cell type, but it's actually still making consistently good accuracy performance. And one thing that's actually quite important here is that we can then interpret the model to understand what exactly the model is doing, right? So even though they're somewhat sort of black box machine learning models, we actually have interpretation methods, which shows that, okay, so if the target sequence has an A at this particular location, that makes it more likely to have insertions, or if it has a G next to the cut site, it's more likely to have deletions, right? So those actually give us quantitative insights. So the thing that's actually quite surprising to us, I just want to share with you, is actually quite a surprising discovery is the following. Right, so I'll just show you this part of the video again, right? So the picture that people typically think about in textbooks is that when you have this double-strand DNA break, right, so you have some local return mechanism, so some local insertions and deletions. Right, so insertions and deletions should be fairly small, right? Maybe one base, you have one base pair or 2 base pairs that are inserted and deleted. What we actually observed in our experiment, which is very surprising to us, is the presence of actually quite long insertions. By which I mean there are actually sequences, oftentimes longer than 20 or 30 base pairs, that are actually getting inserted when I make these cuts. And it turns out that these long insertions are actually quite prevalent in the sense that in over 90% of the locations where we make these edits, we can observe some evidence of these long insertions. And some of them could be quite long. It could be over 50 base pairs. So then the interesting question is, okay, so where are these long insertions coming from and how do they get there, right? Because these long insertions are sort of quite unexpected events, right? That people have not really understood whether they even occur or how they happen, right? But they could have quite important therapeutic implications. So for example, if you want to make an edit at a particular location, you don't want these long insertions to happen. So we did some additional analysis, and it seems like these long insertions are really coming from other chromosomes or from other parts of the genome. Right? Here's just some evidence of this. On the left here, I have sort of this kind of high-C or 3-dimensional contact map of the genome. Right? So this is at a fairly coarse resolution. So each dot here, each square, just show you how often do two different chromosomes interact physically, right? How often do they actually physically touch each other in the nucleus? And on the right plot here is basically how often do we observe these long insertions, right? So that means that if I actually make a cut, if I use CRISPR to make a targeted cut in chromosome 1, for example, how often do I see parts of chromosome 2 or chromosome 10 gets inserted at this location in chromosome 1? And there's actually quite remarkably good agreement between these two plots, right? So this gives us some evidence that for the following mental model, which is that when the CRISPR makes a cut, right, let's say on chromosome 1, now in chromosome 1, there's this double-stranded DNA break, right? And then it's basically the DNA is trying to find some other piece of DNA to insert, right, to put itself back together, to stitch itself together. And sometimes, if there are other DNA sequences that are nearby, then those nearby DNA sequences are more likely to get inserted during this repair process. And if it happens that the nearby DNA sequence actually come from different chromosomes, which is often the case, then that actually also makes it likely for these nearby chromosomes, for these other chromosomes, to get inserted just by chance. Right, so we don't have sort of complete proof for that model, but I think based on the data that we have, seems like one plausible model for how the insertions occur and where they're coming from. So I think going forward would be actually quite important to really understand how these insertions happen, because they could actually really lead to sort of unexpected outcomes when we use CRISPR. So we've also validated and observed these insertions in other cell types in other systems as well. So finally, we also produced this web portal where people can input their own DNA sequences, and then our algorithms sprout to make these different predictions. Okay, so just to summarize this part, right? So hopefully this gives you sort of one example of how we can use machine learning, right, to predict CRISPR outcomes. I think the most interesting application of this machine learning tool is to help us to prioritize and to figure out which part of the human genome is actually safe to make edits and which part is not safe, right? Because the algorithm will tell you that in some parts of the human genome, if you try to make edits there, you're much more likely to have unexpected insertions and deletions and even long-range insertions. Whereas in other parts of the genome, right, the precision is much higher for these repairs. Right? So that actually gives now like a ranking for if we're trying to target a particular gene. Now we know that some parts of the gene are actually safer to make these edits, and other parts of the gene are not so safe. Right? Now then we can design our editors to target the safe locations. I think these long-range insertions are actually really interesting. We don't really exactly know the mechanism by which they get inserted, but I think that's something that's really important to understand better. Okay, so I think this wraps up the first part of my presentation. I want to maybe just see if there are any questions, and then I have like a few more minutes to tell you a bit more about some of the machine learning technologies that we developed. Are there any questions for this first part?
周老师您好，可以问吗？我是杨俊，
Yes, 可以。
上海的杨俊。比较好奇你的这个名字 Sprout 是来源于 Sprouting 吗？还是来源于什么样一个怎么取的这样一个名字
？Oh, yeah. So Sprouts 这个名字的话，就是我这里这个 Sprout 是那个英文就想就是叫 CRISPR， 因为我们这个 algorithm 就是做 CRISPR outcome prediction。Right, so CRISPR outcome 的话，只加在一起的话就变成 sprout。
挺有意思。然后
其实我有两个，
不好意思，一个问题就是刚才提到在 HiC 那部分，我们用到发现它其实这些隐射性可能来源于其他的染色体，然后有没有看这些序列可能是比如说是重复序列，或者是基因区的序列，或者这些序列有没有特性？
对，然后我们看这个序列的话，大部分这个 sequence 都是从 enhancer 出来的，可能很多都是 non-coding regulatory elements。
它的长度足以判别它的准确位置，对吗？
对，这些长度的话，我们主要是看是比较长的 sequence，就是长于有些像这些大部分都是长于50 nucleotide， 所以因为它们比较长，所以我们可以就把它 align back to the genome。
谢谢周老师。
OK，
下面的我 confirm 两个是，我提醒他，我
Yes.
confirm 两个。第一个就是你这里用的那个是 Cas9 吗？就是那个
Cas9， 对。
OK， 这是第一个。第二个就是你谈到的那个 long insertion， 就是在你的这个数据里面有多普遍？因为你前面也有一个展示的那个就是基因 editing event 的那张图，然后其实大部分观测到的应该是一些小的 deletion 或者是一些就是 SNP， 对吧？然后我想知道这个 long insertion 大概占多大的比例？然后这个 observation 是在别的系统里面也经常看到吗？
对 ，OK， 这是非常好的 question。So 这些 long insertion 的话，我们因为我们做了1000多个这个 CRISPR edits 在 T cells， 在这1500多个 CRISPR edits 在 T cell 里面的话，90%以上的话，这个 edits 都可以看到这些 long insertion。但这个的话就是说，more precisely 这个应该说的话就是 in 90% of the edit target site， 我们可以看到long insertion at relatively low frequency, right? So by frequency, it means typically in these cut sites, the frequency would be less than 1%. Right? So it's actually very prevalent in that we can see it in many locations, but at each location, the frequency in the number of cells is actually less than 1% for these long insertions. 然后你第二个问题的话，就是说在其他的 cell type， 我们对这里是我们可以看到像 T cells 或者是其他这个 HEC 或者 K562 就是 different cancer cell line 都可以看到这个 long insertion。
OK， 然后如果再有一个 follow up 问题，我觉得很有意思的是看其他的那个 CRISPR 系统会不会也有类似的情况，像那个 Cas12 还有其他的，就是
对
会比较有意思。
Yeah.
按照这个 hypothesis， 其他的很有可能也会有类似的情况，对吧？
对 ，good question。
OK，
我说一下，我叫陈金峰，是那个做转作子的。然后您说这个现象其实就是说任何 double strand break 修复的时候都会有这种插入的现象，就是它是叫那个failure DNA， 就是说在修复的时候有时候是那个。由于那个 non-homolog end join 它修复的时候不够完美，所以它会随机的产生这样的，就是从其他地方抓一个 DNA 过来填上去。就是说这个现象其实是比较普遍，最早应该是在80年代，然后在转作子跳动的时候发现的，就是后来发现其实它是非常普遍的。就是说，但是在 CRISPR 里面看到这个还是就是说比较意外，就是说这么普遍的话还是比较意外，因为它在我们就是说你应用 CRISPR 的时候，这个是一个非常不好的一个现象，对吧？
对，然后我们主要也是说的话，因为它这个尤其在 CRISPR 里面，它可能是 really 跟这个 nuclear structure、nuclear 3D structure 很相关。
对，就是可能在 CRISPR 系统里面，它的机制可能跟其他的会有点不一样，就是说，但是这个在 double strand break 修复的里面是比较那个常见的。
That's right. Yes.
谢谢啊。
Okay. Great.
好，我有一个问题，
Yes.
可能很外行啊，就是你说这个 3D genome interaction， 但是它 chromosome 的位置，空间上的位置在不同细胞里，在一个细胞在不同时间上，它是稳定的吗？比如说 chromosome 22一直和 chromosome 1 interact 吗？还是怎么样？
这也是一个很好的问题我们这里这个 3D interaction 的话，这是一个 average interaction。像你说的话，在不同的细胞里面，或者一个细胞不同时间里的话，这个 chromosome 都会可能是在有些变动嘛。但是这个的话，这个 experiment 的话，就是用很多这些细胞，然后这 average， 就说这个在一般情况下的话，这个 chromosome 跟这个 chromosome， 或者这one part of one chromosome and this other part of the second chromosome, how often do they interact? keep an average effect.
那它的 sister chromosome 不会，这可能是个非常 naive 的想法，但它 sister chromosome 不会近一点吗？不会，比如说在分裂的时候，不会更容易发生 recombination 或者，我不是特别清楚这个。
Yeah, I think that also depends on which stage of the cell cycle the cell is in. So here we just want to say that yes, there's some non-uniform interactions between different chromosomes, right? Based on which ones tends to be physically closer to other ones. And that non-uniform interaction is also predictive and highly correlated with these long insertions that we observe.
Thank you.
Okay. Yeah, these are all very, very interesting questions. Okay, so for the second part, and then I can go a little bit more, and then I'm happy to stop and take more questions. For the second part, I actually want to tell you about some of the machine learning tools that we developed a little bit more, right? Because I think there's a lot of excitement about, for example, applying deep learning, machine learning to genomics, right? So one of the big major obstacle is really because these tools are fairly, they're very complicated neural networks models. It's actually very hard for researchers, for practitioners to really understand what exactly are these models doing. So we have been working on trying to develop some ways to really, I think, hopefully gives us some more insights that these models are actually not really as complicated as they seem. So I want to just illustrate this through a few examples, right? So we've developed an approach that we're calling neuron Shapley value, right? Which is basically a method that assigns a score, like an importance score, to every neuron in this neural network. Just to make it concrete, right? Suppose I actually take this image and I have a neural network that are used to take the image and try to predict whether it's a volcano or not. Right? So that neural network could have many thousands of neurons. And then our neuron Shapley approach basically then assigns a number, right? It could be 0, or it could be -1, or +10, right, to each of the individual neurons. And the number basically tell you how important is that individual neuron to the performance of this overall neural network. Right. So, and something actually really interesting happened when we computed those importance scores. So it turns out that you could have this neural network that's actually really big, right? Could have tens of thousands of neurons, but only a very small number of these neurons ends up being important for the predictions. Right. So we identified that most of the scores, the Shapley scores, are very close to 0. Right? There's only a small number, like 30 or 40 or 20 neurons that have like substantially non-zero scores. And the reason why we know that they're actually really important is by doing the following kinds of ablation experiments. Right. So what this plot here shows you is that if I take the neurons that have the highest neuron Shapley score, right, I just remove them from the network. And if I, after removing about 15 of these highest scores, the neurons with the highest score from the network, the entire neural network's performance has basically collapsed. Right, so the x-axis of this plot here shows you how many of the neurons have I removed. And the y-axis shows you a metric of the performance of the network. In this case, it's the recall. Right, we can do this for accuracy, for precision, for other things as well. So the red line corresponds to if I just randomly remove neurons from this neural network. Right, so if I just remove 30 random neurons out of the many thousands of neurons, right, so the network really doesn't change. But if I actually remove the neurons that we identified to be the most important neurons, just after even removing 15 of them, the network has completely collapsed. Right? So this suggests that you can have this very complicated neural network, but there's only a small handful of neurons that are really critical for the performance of this network. There's only like 15, 20 neurons that are really important. Right? And that's actually really good for us, for scientists, because then we just have to understand what are these 20 neurons doing, right? So if there are 20,000 neurons, then it'll be really difficult for us to understand, right? But if there are only 20 important neurons, then I can just look at those 20 neurons even individually and then we try to understand what are each of them doing. So for example, just to give you some visualizations, right? One thing we can do is to actually, here is to say, okay, so the single most important neuron, right, for this whole neural network is actually this neuron that's in the 6th layer, in the second branch of the 6th layer, right? And there's just that one neuron there that's really important. And again, here the task, the neural network is actually trained to predict which images are volcanoes or which images have volcanoes in them. And what we can do is that we take this single most important neuron, we can see, okay, what are the images among like all of my image from the Google image, or which image actually gives me the highest activation for this single neuron, right? And which image gives me the lowest activation for this neuron. That's what we show here, right? The highest activation one is in the first row and the lowest activation one on the second row. So it's actually pretty clear then, it seems like this single neuron here is really looking for things that look like clouds, right? So it's basically, there's one neuron in this entire neural network that's looking for clouds. And that ends up being really important for deciding whether this image has a volcano or not, right? And it's also highly unique in that if I just remove this one neuron, there's not another neuron in the network that's looking for clouds, right? So if I just remove this one neuron, the network's performance actually drops quite a bit. So this left plot here is like another way to visualize it, right? So instead of looking at natural images, I can see which synthetic image actually gives me the highest activation for this neuron, right? So here, the synthetic image is also basically looking for these kind of cloud-like shapes. And I can just do this. There's only like 10, 15 important neurons. I can just do this one by one. So it seems like the second most important neuron, the one with the second highest value, is on the next layer, in the 7th layer, right? And it's basically looking for like kind of mountains, right? So there's one neuron that's looking for cloud, one mountain that's looking for mountains. And if we're missing these two neurons, the entire network just stops working. And we can do this, so this is a general method that we've developed called neuron Shapley. We can do this for other neural networks, for other prediction tasks. So here I'm showing you a few more examples. For example, here you have a neural network that's trained to predict whether there's a dumbbell in the image or not, right? And for that dumbbell predictor network, we are showing you two of the most important neurons, right? Seems like one important neuron is basically looking for like a dumbbell shape, and the second important neuron for this predictor is actually looking for human arms, right? So it's basically learned that most dumbbell images will have actually some human holding that dumbbell. That's why it's looking for arms. And it seems like there are only these two neurons. One is looking for the dumbbell shape, one is looking for the arms. That makes this predictor effective. So what we're interested in doing now is now that we have this general technology for neuron Shapley values for identifying which neurons are really important in the network. We can also now go back to apply that to even biological settings, right? For example, people are now training neural networks to predict breast cancer from mammography images, right? So those are big neural networks, but we can then apply this kind of neuron Shapley technology to figure out what are the small number of neurons that are actually important in that whole neural network, and then that enables us to understand what is the network doing. Okay. And we've done this now for a variety of different neural networks, and it's actually quite interesting to understand which layers of the network tends to have these really important neurons. Okay, so the last thing I want to quickly mention is that one of the big things about, since we work in deep learning, developing machine learning algorithms, right? So if we really want to deploy these algorithms, especially in medical and clinical applications, one of the big challenges is that the clinicians and our collaborators in the hospitals, they're not really computer scientists, so it's actually oftentimes hard for them to interact with these models. So we actually developed a tool called Gradio Hub, which is a platform that hosts these models, right? So that people can interact with the models in real time. So if you're a machine learning developer, the way that you can use Gradio is that you develop your machine learning model, and then you can just add 2 lines of Python code, and Gradio will actually create an interactive user interface. It also creates actually a link, like an example link, where you can actually share that link with your collaborators, and your collaborators open up that link, maybe then they'll actually be able to interactively interact with your machine learning model in real time, right? And they can get feedback in real time. So this, I think, is hopefully a tool that will be useful for the broader community that makes it easy for even non-specialists to interact and provide feedback on these machine learning models. I think with that, I'll stop here. This is the end of the presentation. I'm happy to take some more questions and have additional discussions.
Hey, James, 我有一个问题，就是是针对你的第一部分的关于 CRISPR 预测的那一部分，然后我想问一下，就是在人类里边，比如说一些大的 structure variation， 像 inversion 啊，translocation 这些，然后对你的那个 model 有多大的影响？谢谢。
Yeah, that's also a good question. So for our model, it's not too affected by these large structural variation, right? So it's really, it seems like it really uses the more local information, like just like local, a few base pairs of DNA sequence. That seems to be the most important features. But certainly like the long insertions that we discovered in our experiments, those could be indicative of larger structural variation that are caused by CRISPR, right? So that's something that we would like to understand better.
谢谢哦。
James 非常感谢你的报告。然后关于你第二部分讲的，尤其是那个 neural shuffling 的那个 methods， 你能不能在生物学的背景下讲一下，如果我们有 training 这样 deep learning model 的话，怎么样理解这个结果呢？
Yeah. So, for example, here I showed you some 就是 image example, right? Image 的这种例子，就是我们可以说，我讲的第一部分就是 CRISPR， 就是这做一个例子。假如说我可以用 CRISPR， 就是当于 training data, right? 然后我也可以 train 一个 neural network to do CRISPR outcome prediction， 就是 input 的话就是 DNA sequence， 然后 output 的话就是这 CRISPR prediction。然后用这个 network 的话，我也可以用这个 neural Shapley。neural Shapley 就告诉我这个 network 的 individual neuron， 就是 which neuron actually are the most important for this prediction, right? And what we find here is that only a very small number of neurons, right, are really important. Right? So in the CRISPR example, we can then see, okay, what are these neurons looking at in the DNA sequence that are, that leads to the CRISPR predictions.
对，我觉得我的问题是怎么把这个跟生物学联系起来，就是哪些我们这是可以通过图片，然后我们找到这个 major feature， 然后这个 feature 你可以 reconstruct 这个 feature， 然后
yes，
但是在 sequence 背景下，这个 sequence 的 combination 回来的 information 基本上不能告诉我们任何的，或者很难告诉我们任何的 biology 的问题。
Yes, so I think there are some methods that are related, right? So in this example here, I show like two methods to figure out what this neuron is doing, right? We said that this is a really important neuron, and one thing we can do is to see which natural input in my data set gives the highest activation, right? So I can do the same thing with that in the CRISPR case too, right? So I can then see, okay, which CRISPR target sequence, right, or subsequence actually gives me the highest activation for that neuron. And that subsequence then becomes sort of like a motif, right? It could become, for example, binding sites for transcription factors, or some sort of microhomology that's useful for this DNA repair process, right? So that's one way we can get some of the biology insights from these models.
就是说，你这 model 可以回来再接着返回一些，如果在 biological setting 下也是能够返回一些 sequence， 就某个 neuron 可以从 reconstruct 它的 sequence 的 base pairs 是吗？
That's right. Yes. 对。
Oh, that's awesome. Cool. 谢谢。
我可以问个可能比较外行的问题，就是 如果你有那个 neural Shapley values， 然后说其实整个 neural network train 好了以后，只有几个 neuron 是，几十个 neuron 是特别重要的。那你能不能说据此把这个 neuron 简化成一个小的多的 model， 这样子就可以加速做大规模的运算，可能会容易一点。
对，这个也是一个很好的主意。然后这个也是这个一个 application， 这个 application 好像是用这个 pruning， 因为有一些这个 network 的话，它本来就是比较大嘛，所以它用的这些能源啊、时间呢比较长。我们现在的话知道这个 network 不需要这么多 neuron， 只是只有几十个 neuron 是最重要的啊，其他很多 neuron 的话，你都可以把它扔掉，然后这 network 的话，它 performance 也是差不多跟以前，所以这个的话也是一个 application 用来 reduce the size of the neural network。
好，对，谢谢。
周老师你好，上海杨俊，再想问一个问题，就是关于这 neural network 会不会那些小的 neuron 是防止这个模型犯一些低级的错误的这种概念？有没有这种可能？
对，然后有一些 neuron 的话，我觉得我们 yes 我们发现的话也是有 about 20-30 neurons that are quite important for preventing the neural network from making these like adversarial so like very bad mistakes. So there's a small number of neurons that are useful for preventing those mistakes.
James，
我接着问一个金良刚才那个问，就是这个一个是可解释性，另外就是关键的 feature， 然后还回到我们讨论的 CRISPR 那个地方，然后在你构建你的 machine learning model 的时候，其实你是有4个 feature， 一个是 DNA sequence， 一个是那个 chromatin information， 然后还有那个 thermodynamics， 还有那个 expression。然后你们建完这个模型有没有看这4个 feature 里面哪一个对模型最后的结果贡献最大？然后是不是这4个都是 essential 的，就是在预测这个 CRISPR 基因outcome 的时候。
Okay, great. 对，然后这个各种各样的 feature 这里边的话，还是 DNA sequence 这个 feature 是最重要的。然后其他的这种 feature 的话，我们可以是它们的效果还是比较小的 compared to DNA sequence。然后 DNA sequence 里面的话，也主要就是 very nearby sequence， 也不需要看得很远，就是最近的几个 nucleotide 可能是这是最重要的。
好，谢谢。
James， 我想问一下，就是在你这个 machine learning 那个轴的另一边，关于那个 text processing， 就是相当于 natural language processing 这方面，如果我非常感兴趣的话，有没有可以介绍的这个资源来学习？特别是对于像中文这样子，就是很特殊的字符，那现在就是在世界上，他们对这种language processing 的那个研究处在一个什么样的？能不能给我介绍一下
Okay, yes, yeah. 然后 natural language processing 像 NLP 现在也有一些比较好的像 online class， 像 Stanford 的话有一个也是就是 publicly available， 这是可能放在 YouTube 上面，这叫就是 natural language processing 的 class， 就是用 deep learning 做 natural language processing。然后这个的话也是，而且 it should be a good resource and good reference。从中文像 Chinese 来说的话，现在因为那个也有很多很好的research 是从一些 Chinese companies， 像 Tencent、Baidu， they also have very strong NLP research groups. So they've actually published some very strong models. Also, Google has also some good models that are designed specifically for Chinese language. But I think the Chinese companies like Tencent, Baidu, I think they have the best models for NLP models for Chinese.
大家还有问题吗？ James，不知道你有没有兴趣或者有没有时间讲一下你提到的关于 COVID 建模的一些研究。
Yeah. I think it's certainly still very much ongoing. But one of the things that we're interested in doing is to model the spread of COVID-19. I think one of the big challenge there for modeling for prediction is to quantify in the general public, right, how many people have been infected but have not been actually confirmed or even tested. And because especially in the US, the amount of testing is actually quite low. So there could be a lot more people who are infected out there now, but who have not been tested. Right. So one of the things we're thinking and working on is to develop time series models to model the number of infected individuals. Right. So the other thing that I think is quite important and interesting to predict and to model is to predict what are the effects of different interventions. Right. So currently, for example, in the US, there's a lot of, and also in China before, right? So there's policies to encourage people to stay home, to not go outside, right? It would be actually really useful to have more quantitative models to show how, like, what is the actual effect of such policies, right? If people are encouraged to really stay home, does that really, to what extent does that really decrease the spread of these viruses? And that's where also I think some of the machine learning becomes quite useful, right? Because to really model those things, you want to understand, and in normal times, right, when there's no virus, how often do people come into contact with each other? You can do that by analyzing, for example, social networks or location data. And then you want to see when there is this policy for people to stay at home, how does that actually change people's behavior, right? You can also try to use location data, social networks to see how the behaviors change. And from that, you can try to then have some estimates and models and predictions for how does that actually, to what extent do these kind of policies are effective in reducing the spread of the virus. So those are two areas that I'm quite interested in.
Sounds very interesting. Any more questions? All right, thanks, James, and thank you so much for giving the talk, and it's very interesting, and hope to talk to you later more about, hope to keep in contact, and thank you so much for joining our online seminar.
Yeah, well, thank you very much for organizing, and if you have questions, feel free to contact me. I'm happy to follow up.
Thank you.
非常感谢 James， 谢谢你就是抽时间过来给我们讲这个。
Yeah， 非常高兴有这个机会来 ，OK， 可以保持联系。
好，
好，
今天就暂时到这里。
Thank you.
Okay.
Have a good night.
Good night. Take care.
再见。
Bye. Keep healthy.
Bye.
Yeah， 再见。
再见。